Hello students. In this session, we are going to solve test paper that is AIET 3 test paper, which has the code that is YX3723 for class 12. So here are some of the difficult questions that we picked up and the important questions as well. So let us solve one question after another and let's see if you guys uh, got this right or if you have solved your doubts. Okay. So here is your first question. So that is question number 104. Which of the following is true about purine and pyrimidine bases? And our options here, right? So our options are they are hydrophobic and relatively insoluble in water at the near neutral pH of the cell. At acidic or alkaline pH, the bases become charged and their solubility in water decreases. Purines have two rings in their structure, but pyrimidine bases have only one ring. At acidic or alkaline pH, the bases become charged and their solubility increases. So we have the options here that is A, B, C, D, which says A says 1, 2, and 4, B says 2 and 4, and C says 1 and 4, and then D says 1, 3, and 4. So when we look at this question, so let us see which of the following is true with respect to purine and pyrimidine. So let us look at the first option, that is they are hydrophobic and relatively insoluble in water. So what we're talking about here is purines and pyrimidines, that is adenine, guanine, cytosine, and thymine, which are the nitrogenous bases that we see in DNA. Okay. So the option that is they're hydrophobic and relatively insoluble in water at near neutral pH of the cell. So this is absolutely right. Okay. So they are hydrophobic and they are insoluble, especially in the neutral pH. That means you know the pH of water, right? 7. So at that pH, so you can find these nitrogenous bases acting as insoluble in water. So that is correct actually. Coming to the second option, that is at acidic or alkaline pH, the bases become charged and their solubility in water decreases. So this option is particularly wrong. Why it is wrong? Because at acidic or alkaline pH, what happens to these bases are they become charged and their solubility in water increases, so not decreases. So this second option becomes wrong for us. Okay. And coming to the third option, purines have two rings and in their structure, but pyrimidines have only one ring. So this is what the chemical structure or the molecular structure of the nitrogen bases, which helps us identify these bases, right? So whenever it comes to the purines, so purines, we see that their structure is particularly something like this, right? So we can find them having two rings. So in one ring, you have six atoms. In another ring, we have five atoms. So the six atom ring is called as purine ring, right? And then the five atom ring is called as imidazole ring. So basically what it says is, whenever you have a molecular structure with two different rings, they are called as heterocyclic compounds. Okay, hetero means you know that it's different, and cyclic means two different atoms are there in a cyclic structure. So in the six atoms of the purine, we have around four nitrogen and two carbon atoms. Here we have again three nitrogen and two carbon atom. Okay, so that is why this option is correct. That is the third option where purines have two rings in the structure. Example is adenine and guanine for purines, whereas in pyrimidines, we have only one ring, that is uracil, thymine, and cytosine are pyrimidines. So this option is correct. At acidic or alkaline pH, the bases become charged and the solubility in water increases. So this option is right as I explained it already. So for 104, the correct option is option A. Okay, so let's go for the next question. If Henle's loop were absent from the mammalian nephrons, which one of the following is to be expected? There will be no urine formation. There will be hardly any change in the quality of urine formation. The urine will be more concentrated. The urine will be more diluted. Okay. So let us see what is the function of the Henle's loop. Actually, if you see, the loop of Henle in mammalian nephrons are very, very large or bigger compared to any other organism. Okay. In especially mammals, it's very big. So what happens in loop of Henle? So you can see, guys, like if you see nephrons, we have something called as glomerulus, right? And after that, we have proximal convoluted tubule. So there it leads to our descending loop of Henle, and then we have ascending loop of Henle. So it is associated with our vasa recta. So what is happening here? So the descending and ascending loop of Henle, and then it will lead to distal convoluted tubule. Anyways, so here the role of your glomerulus, the role of your PCT, and the role of your descending loop or ascending loop is to concentrate urine. So what is happening here? The ascending loop and the descending loop, they are involved in something called as counter current mechanism or counter current multiplier system, what we call it as. So in this counter current multiplier system or counter current mechanism, the concentrating of urine will take place. Okay. So usually what happens is the water, excessive water that is retained in the system or whatever the filtrate, what we call it as. So in this filtrate, the water is removed and the concentrated urine is formed. So if the loop of Henle is removed, the reabsorption process and the ultrafiltration process, what happens is basically broken down. So whenever the reabsorption process is broken down, what will happen? What is reabsorbed? Basically the important minerals as well as the important water and glucose is reabsorbed, right? So if that part which is involved in this process is removed, what will happen to the urine? So there will be just the water keeps on getting wasted in from our body as it enters the kidney. So the urine will have lots and lots of amount of water, lots and lots of amount of important salts like sodium and potassium. Okay. So moreover, the urine will look more dilute. So our option B is right for 107. Question number 107. Now, we have the 109 question. It's very, very interesting. And it's easy also. Okay. In fruit flies, dumpy wings are shorter and broader than normal wings. Okay. The allele for normal wings is D, is for dominant to the allele. For dumpy wings, that is small d. Normal wing flies were mated and produced 300 normal wing and 100 dumpy wing flies. The parents were probable. So if you just observe this, they're talking about 3 is to 1 ratio of phenotype. So where can you observe this 3 is to 1 phenotypic ratio? If you observe this, you can think about, okay, we might have seen this somewhere in our monohybrid cross when we talked about tall and dwarf plant. Similar, right? So what is happening here? If we had taken, when we took in the F2 generation, that is, I will compare it, okay? So capital T, small t was crossed with capital T and small t. What was the outcome? Capital T, capital T, capital T, small t, capital T, small t, and small t and small t was the outcome in the F2 generation. Here, what happened? Three of the offsprings are actually tall and one is dwarf. So one is dwarf, right? So what is the ratio here, students? That is 3 is to 1. So this is a phenotypic ratio. So here also what they're talking is, we are talking about this question, that is Dross of Lamb, where we have a dominant D, if it is crossed by, let us call it as a heterozygous individual, okay, with a normal character. See, normal wings are crossed, right? So when they're crossed, what will happen? We'll get capital D, capital D, capital D, small d, capital D, small d, and small d, and small d. So what they're telling here, we have 300 normal winged. Of course, this will give rise to normal wing. This will give rise to normal wing. Why this will give rise to normal wing, students? So we know that capital D is dominant over small d. So according to the law of dominance, this should come only the dominant characters will be expressed. This also will be normal winged. So three of them are normal wing. That means if 400 are born, 300 will be normal wing, and small d and small d will be the dwarf wing or dumpy wings. Okay, so that will give us 100 offsprings. So our option for this is only this will happen when a capital D small d is crossed with capital D small d. Okay, so that is why for 109 our option is C. All right. Okay, students, let's go for the next question. Read the following 5 statements 1 to 5 and select the option with all correct statements. Mosses and lichens are the first organism to colonize a bare rock. Salaginella is a homosporous pteridophyte. Okay, colloid roots in cycas have WAM. And then main plant body in bryophytes is gametophytic, whereas pteridophytes, it is sporophytic. In gymnosperms, male and female gametophytes are present within the sporangia located on the sporophyte. Okay. So let us see which are the right options. Okay, we have to select the correct options. Correct. So the first option, mosses and lichens are the first organisms to colonize a bare rock. So this is very, very true. So there are many forms of lichens. For example, fruitiform lichens, and then we have cruciform lichens. So here there are specific type of lichens where they actually they grow on bare rocks. If you just see in the mountain sides when you go, you can see there are greeny patches on the surface of the rocks. So these green patches looks alive and they look leafy also. So they are nothing but our mosses or lichens which are growing on the rock. So they are the first to colonize the bare rock, and they even have the properties to break down the rock particles and convert it to a fine sand. So which are the colonizers. So the first option is right. Now, Selaginella is a homosporous pteridophyte. It is actually a heterosporous pteridophyte. Now, this is wrong. And then colloid roots in cycas have VAM. So basically, a VAM is a mycorrhiza. Okay. So the roots of the cycas is colonized by not VAM. Okay. Instead, it is colonized by blue-green algae such as nostoc, anabena, okay, etc. So that is what colorite roots in cycas are symbiotic with. So they're symbiotic with blue-green algae, not with VAM. Okay, VAM refers to your mycorrhiza, M for mycorrhiza. Okay, so that is why option 3 is wrong. So coming to the fourth option, that is main plant body in the bryophyte is gametophytic. Okay, they're telling that the main plant body is gametophytic, right? True, because they are haploid. Whereas that in pteridophytes, it is sporophytic. Yes, the dominant body, plant body in bryophytes is basically haplontic life, that is gametophytic, whereas in pteridophytes, it is sporophytic, right? So coming to the fifth option, that is in gymnosperms, male and female gametophytes are present within the sporangia located in the sporophyte. This is very true. In gymnosperms, what happens? The main plant body is a sporophyte, and sporophyte bears sporangia, and within the sporangia, you have your male and female gametophytes. So in short terms, we can just conclude that in gymnosperms, the gametophytic life is absolutely reduced. Correct, right? So once again, yeah. So coming to yeah, what is the right option for 116? 116, it is actually A. Okay, 1, 4, and 5 are the correct options. Okay, so let's go for the next question. So hold on. Okay, so coming to the next question, which is, yeah, 118. Which of the following statements is or are true, are correct events in aerobic respiration? Okay, here are the options. The complete oxidation of pyruvate by the stepwise removal of all the hydrogen atoms, leaving 3 molecules of oxygen. Okay. Complete oxidation of pyruvate by the stepwise removal of all hydrogen atoms, leaving 3 molecules of carbon dioxide. The passing of the electrons removed as a part of a hydrogen atoms to molecular, which stimulates a synthesis of ATP. The passing of the electrons removed as a part of hydrogen molecules, okay, to hydrogen molecules, which stimulate the synthesis of ADP. So we have to choose which is the correct events with respect to aerobic respiration. So let us see the first option. So when we look at our first option, that is the complete oxidation of pyruvate by the stepwise removal of hydrogen leaves raised to 3 molecules of oxygen. This is absolutely wrong. Why? Because the stepwise removal of hydrogen from the pyruvate would lead to the release of 3 molecules of carbon dioxide. So our option 2 becomes right. Okay. So coming to our option 3, that is the passing of the electrons, removed as a part of hydrogen atoms. So they also produce, they also help in synthesis of ATP. So this is all right. Okay, this is right. So whenever the hydrogen electrons are removed, so as a part of electron transport chain, what do we get? We synthesize ATP, right? So that is why this is correct. Whereas the last option, it is not ADP, it is given. The third option is right. So our 1 and 3 are correct, which is 118 option. Sorry, 2 and 3 are correct. That is why our option C is correct. Okay. So let's go for the next question. Which of the following statements 1 to 4 are correct? In elution, the separated bands of the DNA are cut from the agarose gel and extracted from the gel piece. Okay. Next one. E. coli cloning vector, that is PBR322, shows several restriction ORI antibiotic resistance genes and ROP. The downstream processing and quality control testing vary from product to product. Next one, complete bacterial cell cannot be, sorry, competent bacterial cell cannot take up plasmid. Okay, so we have the option all of the above, none of the above, 1, 2, and 3, and D says it's only 4. Okay, so question number 122, let's see which is our right option. So coming to our first option, that is in elution. So elution step, we know that in agarose gel electrophoresis, so the bands are separated based on the size. Once the bands are separated, so we can easily measure the size of the bands on the distance they have traveled, and we can see based on the hybridization that we can isolate this band and it can be extracted. Okay, we can extract a piece of the DNA fragment from the separated bands. So the first option is correct. Coming to the second option, yes. What makes E. coli's PBR322 an ideal vector? Because it has ORI site, it has restriction site, it has selectable markers like antibiotic resistance gene and as well as ROP. So that is why PBR322 is an ideal vector. So this option is also right. The downstream processing, okay, and quality control testing vary from product to product. Absolutely true. If you want to purify enzymes, if you want to purify proteins, if you want to purify any of the bioactive substances, okay, so for everything, based on the chemical nature, based on the organism used, based on the procedure, based on the bioengineering, so everything varies, right? For example, if you take the difference in the process between the insulin production and distillation of alcoholic drinks like beer or anything. So you can see there is a plenty of difference between how the downstream processing takes place by the time the product reaches us. So this also is a very right option. Now, the competent bacteria cannot take up the plasmid. So basically, competent bacteria are the ones which take up the plasmid. So what is making the bacteria competent? Competent, we do it several times by adding calcium, basically we add calcium, right? So when we add calcium, bacteria competes for the DNA that is in the surrounding and it takes up. So our option 4 look wrong. So according to the question, 1, 2, and 3 are right. So option C is right for 122 question. So we have 135th question. In a seed of maize, scutellum is considered as cotyledon because it. So we have the options: protects the embryo, contains food for the embryo, absorbs food materials and supplies them to the embryo, converts itself into a monocot leaf. So from the given options, why is scutellum in the monocot seed considered as a cotyledon? So if you just consider the monocot seed, so what is a scutellum? If you just want to remember through visualization, you can see here, this is the endosperm, right? So, endosperm. And then here we have coleoptile and coleorhiza, plumule radical. And here is where your scutellum comes. So scutellum, basically what it does is it absorbs the food materials from the endosperm region and it supplies to the development of embryo. That is why it is called as a cotyledon. Okay, so our option C, that is absorb food materials and supplies them to the embryo, that is absolutely right. Okay, 135 is C. Next question. The shifting of oxyhemoglobin dissociation curve to the right due to increased concentration in blood is also called as, okay, Hamburger's phenomena, and then we have Bohr's effect, we have chloride shift, and then we have Donan equilibrium. So what happens? So they're telling about this shift of oxyhemoglobin's dissociation curve towards the right side. See, there is an effect called as Bohr's effect. So what happens in Bohr's effect is the affinity of the hemoglobin towards the oxygen, it is lost, and it makes, and the tissues make the, and basically what happens, the oxygen from the tissues are released. So this effect, where the hemoglobin loses the affinity for oxygen, so that moment what will happen, the oxyhemoglobin curve shifts towards the right. Okay, so that is called as Bohr's effect. And usually what is chloride shift is, basically you have in your blood, we have chlorine molecules, right? We have chlorine. So chlorine is basically negatively charged. And we know that whenever we have HCO3, that is, the bicarbonate forms of carbon dioxide, right? So we have carbonic acid. The carbonic acid is split into hydrogen atom and bicarbonate. So now what happens is there is an accumulation of your, accumulation of your bicarbonate around the red blood cells. So now what happens, the chlorine molecules will take their place, and now the bicarbonate will come away from the surrounding of the red blood cells. So this is called as a chloride shifter. Okay, whereas a 2-1 is not so related here. So let's quit it. So according to the question 138, whenever the oxyhemoglobin dissociation curve shifts to the right, okay, so that is called as a Bohr's effect. That means there is a decrease in affinity of hemoglobin towards the oxygen. Next one. If there are 1,280 microspores in a tetralocular anther, how many microspore mother cells will be there in each pollen chamber? So 80, 160, 240, 1,280 are our options. So let us see. The question says there are around 1,280 pollen grains, okay, in an anther, and they are specifically mentioning that there are tetralocular. That means there are 4 chambers. That is what the tetralocular anther is. Now, what they're telling is 1,280. If there are these many pollen grains in the whole anther, let us see how many we have in each of the locule or each of the chamber. Okay, so it's divided by 4. So now 4 3s are 2 and 320. Correct. So each of them should have around 320 pollen grains. Now, according to our math, we know that each, right, each microspore mother cell, okay, MMC, right, or the pollen mother cell, what we call it as, it will divide, one cell will divide, one cell will divide to give rise to 4 cells. That is 4 pollen like 1 pollen tetrad or 4 pollen grains. If 1 MMC will give us 4 pollen. So then how many MMC is required to give rise to 320 pollen? So again, we divide 320 divided by 4. So that will give us 80. So 80 microspore mother cell should be there in one chamber, right? One chamber. So that is why our option A is correct with respect to question number 145. All right, students, I hope you understood. Next one. Pick out the correct statement. Our first option says hemophilia is a sex-linked recessive disease. Down syndrome is due to aneuploidy. And then phenylketonuria is an autosomal dominant gene disorder. The same option, right? Phenylketonuria is an autosomal recessive gene disorder. Sickle cell anemia is an X-linked recessive gene disorder. So let us see. We have the option that is A, B, C, D. So let's see which of the following are right. So coming to the first option, that is hemophilia is a sex-linked recessive disease. This is absolutely right. So what is hemophilia? We know that, students, that in the hemophilia is actually the hemophilic gene is present on the X chromosome. That is why it's called as sex-linked, and it is of recessive nature. So the person who has this disease lacks the capacity to clot the blood, And we know that the clotting factor is basically anti-hemolytic factor, which is actually factor 8, factor 9, and factor 11. So basically, when they get mutated, it leads to hemophilia, and the person loses the capacity to clot the disease. And the nature of this genetic disease is basically sex-linked recessive disorder. So that is why our option 1 is correct. Coming to our second option, that is Down syndrome is due to aneuploidy. So what is Down syndrome? So Down syndrome, we know that basically in Langdon Down discovered this disease, right? So we know that we have a trisomy in the 21st chromosome. So that is nothing but the aneuploidy, that is a unusual number of chromosomes. Okay. So that is why this Down syndrome is a chromosomal, also an example for the chromosomal disorder. So option 2 is also correct, right? So next one. So coming to the third option, that is phenylketonuria is an autosomal dominant genetic disorder. This is wrong. So basically, it is a recessive genetic disorder. So we know that the person suffering from phenylketonuria has a gene in the autosomes. So basically, what happens? This particular gene is responsible for producing an enzyme where it helps in converting phenylalanine into phenyltyrosine, right? So when this enzyme, that is this particular enzyme is mutated, so now what happens? Phenylketonuria actually increases due to the deposition of phenylalanine keeps on increasing, right? So that is actually a recessive genetic disorder. So option 4 is correct. Okay. Now, sickle cell anemia is X-linked recessive genetic disorder. Absolutely not. Sickle cell anemia is basically an autosome-linked recessive disorder. So here in sickle cell anemia, what happens is it's because of the point mutation of a hemoglobin beta globulin chain in the hemoglobin molecule. So here it leads to the change in the shape of the red blood cells because of the mutation in the beta globulin chain due to low oxygen tension. So that is why our option 5 is also wrong. Okay, so option D, that is 1, 3, and 4 are correct. Okay, so let's go to the next question. Given below are 4 statements, 1 to 4, regarding human blood circulatory system. Okay, so our first option is arteries are thick-walled and have narrow lumen as compared to veins. Angina is an acute chest pain when the blood circulation to the brain is reduced. Persons with blood group AB can donate blood to any person with any group under ABO system. Calcium ions play a very important role in blood clotting. Which two of the above statements are correct? Now we have the options 1, 4, 1, 2, and 2, 3, and 3 and 4 from the respective options. So let us see what is our right option with respect to 159. So we have to find which of the following statements are actually correct with respect to human circulatory system, right? So 159, let's go. So arteries are thick-walled and have narrow lumen compared to veins. That is absolutely right because they carry the blood from the heart, right? So that is why they have to be thicker because the pressure is comparatively higher on arteries than veins because veins it withdraws blood from the body towards the heart. So here it goes from the heart towards the body. So the pressure taken by the arteries are high. That's why they should be having a thick wall. Angina is an acute chest pain when the blood circulation to the brain is reduced. So actually, angina is an acute chest pain. Yes, correct. When the blood circulation to the heart is reduced. Okay, so it is actually heart, not brain. So that is why our option 2 is wrong. Person with blood group AB can donate blood to any person with any blood group under ABO system. So basically, the AB blood group are called as universal acceptors because they have both antigens and they do not form any of the immune responses whenever a wrong blood group is given. For example, you can give AB blood group A or B or even O can be given to the AB blood group. That's why they're called as universal acceptors. If any of the person receives AB blood group, like if a person having A blood group receives the blood from AB, or if a B blood group person receives the blood from AB, or O receives the blood from AB, so that leads to deglutination reaction because of the presence of an foreign antigen, either it is A or B with respect to other people. So that is why this option is wrong. Calcium plays a very important role in clotting mechanism. Yes, this is very, very true because we know that clotting is a very elaborate mechanism, right? So in clotting, we have three processes. So one of the processes is coagulation of blood. In coagulation of blood, we know that it is a cascade mechanism involving around 13 factors of clotting mechanism, clotting factors. So factor four of the blood clotting mechanism or coagulation is calcium. So calcium is very, very important for the pathways to be accomplished so that our fibrin is produced. So that is why our option 4 is correct. So according to this, 1 and 4 is correct. So our option A is correct for 159 question. So here we have the next question, 161. Select the two correct statements out of the four given below. Glucose or galactose may bind with the repressor and inactivate it. In the absence of lactose, the repressor binds with the operator region. Next, the Z gene codes for permease. This was elucidated by Jacob and Monod. That is Frankus Jacob and Jackman Monod. Okay, Jackie Monod. So let's see what is right for 161. So glucose or galactose may bind with the repressor and inactivate it. So we know that it is wrong. So whenever we studied the Lac-Operon model, we know that allolactose goes and binds to the repressor and it changes its molecular configuration, and then the repressor becomes useless, and because it can no more recognize the operator region. So if you still have doubt what we are talking about, so this is what we are talking about is the Lac-Operon model. So we know that in Lac-Operon model, this is how the gene of the Lac gene looks like. So this is promoter, right? So this is operator, right? So this is Lac Z, Lac Y, and Lac A. So now we know that the promoter operator region is blocked by the repressor. So repressor, basically what happens, allolactose, okay? Allolactose comes and binds to the repressor and makes the repressor no more recognize this operator region. So that's why the repressor will leave the operator region free so that the gene can function. It's not glucose and galactose, so it is wrong. In the absence of lactose, okay, so in the absence of lactose, the repressor will bind with the operator region. So whenever it comes to the absence of lactose, so whenever there is no lactose present in the environment, so let's say there is glucose and no lactose, the allolactose will leave the repressor. So when it leaves the repressor, now the repressor can recognize the operator region again, and it goes and binds the operator region and blocks the transcription. Understood? The Z gene, so this option is correct. Okay. The Z genes codes for permease. So let's see. So the Z gene codes for beta galactosidase. No. So that is why this option is wrong. Very important. So the Z gene, which produces beta galactosidase, will help us convert our lactose, which is present in the environment or which is taken up by the environment, into glucose and galactose. Okay. So that is why this option is also wrong. So basically the Lac-Operon model or the understanding of gene regulation in prokaryotes was first elucidated by this Jacob and Monod. So that is absolutely true. Highly regarded scientists. So option 2 and 4, the statement 2 and 4 are right. So 2 and 4, where do we have? Option C. Yeah. 161 option C is right, students. So let's go for the next question. In angiosperms, the functional megaspore in the linear tetrad is generally a micropylar cell, second from the micropylar end, third from the micropylar end, fourth from the micropylar. Okay. So the option for this, correct option for this is actually fourth from the micropylar end. Okay. So we know that whenever the megaspore mother cell divides and she produces 4 megaspores, out of the 3 will degenerate and 1 will become as the functional one, right? So this is the fourth one. Okay, 1, 2, 3, fourth one. So this fourth one will become the functional megaspore, especially when it comes to the formation of embryo sac, right? So this one is correct. Option D is correct for 164 students. Yes, interesting question. Kidney helps in the conversion. Question number 170 students. Kidney helps in the conversion of useful materials and excretes and excretion of waste, and therefore they receive up to 20%. Okay, it's actually 20% here. Okay, 20% of the heart's output of blood, as much as the heart and the brain combined. Which of the following is not reabsorbed actively? Okay, it's not reabsorbed actively by the nephron. Our options are amino acid, urea, glucose, water. So here are our options. So let's see what is our correct option with respect to question number 170. Even though the facts that are given in this question are absolutely right, that kidney receives around 20% of the cardiac output, because kidneys have a major responsibility, especially to remove the toxins or unwanted waste, that is nitrogenous waste from the blood, to keep our body highly detoxic, okay, or non-toxic. So here, when we look at the function of kidney, there is passive absorption and there is active absorption. So especially glucose and water, and whenever it comes to amino acids. See, glucose and potassium and sodium and amino acids are actively absorbed by our kidneys. So have you seen any healthy person, vomiting or excreting any of the glucose through urine? It is not possible. Why? Because 100% of the glucose is 100% of the glucose is actively absorbed by our kidney. Okay, actively absorbed by the kidney. Or make this statement. Similarly, water is very, very essential to maintain the osmotic balance, and amino acids are very, very important because for the construction of proteins. So whenever the amino acids are broken down during the metabolic process, they produce nitrogenous waste, and one of the major nitrogenous waste is urea. So urea is not actively reabsorbed; they are passively absorbed, and they're basically excreted through our system. So our option, okay, our option B, okay, which of the following is not reabsorbed? Option B is urea, so that is not actively reabsorbed. Okay, so option B is correct. So let's go for the next question, students. Which of the following statements are false or true? Calcitonin regulates the metabolism of calcium. Oxytocin stimulates the contraction of uterine muscles during birth. Graves' disease is caused by the malfunctioning of adrenal gland. ADH stimulates the absorption of water and increases the urine production. So here are our options, and options are very nice. A, B, C are true. Only D is false. Only A is true. B, C, and D are false. A and D are false. B and C are true. A and B are true. And C and D are false. So these are our options. Okay. So let's see one by one. So we have to see which is true, which is false. Calcitonin regulates the metabolism of calcium. Absolutely true. Okay, so they actually help in reabsorption of calcium from the intestine, reabsorption of calcium from the absorption of calcium from the intestine, and as well as from the renal tubules, right? So calcium is absorbed from the blood so that they preserve oxygen within the system. Oxytocin stimulates contraction of uterine muscles during birth. Yes, so this is also true. So basically what happens, you know that it is a positive feedback mechanism during the childbirth. Whenever there is oxytocin that is released by our pituitary, more and more oxytocin is released as the contraction increases by the uterine muscles. So it helps in parturition process, that is pushing the baby through the birth canal so that the baby can be delivered. Okay, next one. Graves' disease is caused by the malfunctioning of adrenal gland. This is false. Actually, it is thyroid gland. So basically, Graves' disease occurs when there is a malfunctioning in the thyroid gland. Next one. ADH stimulates the absorption of water and increases the urine production. So now what happens is ADH, that is antidiuretic hormone, is a vasoconstrictor. Vasoconstrictor means it do not allow sufficient amount of blood to pass through your kidney. So we know that as the amount of blood passes through your kidney, the more, that means the more water have to be removed, right? If your body is dehydrating, if your body should retain water, then what will happen? The blood vessels are constricted so that less blood supply happens to your kidneys, and so that the less water is excreted out of your system. So here is the opposite option that is given. That is, it stimulates the absorption of water. That is true, and increases the urine production. Actually, it decreases the urine production. So that is why it is false. Okay. So true, true, false, false. Where do we have? We have A and B are true, and C and D are false. So option D is right for 178th question. So let's go for the next question. What is a disadvantage of using, okay, protein insulin from pigs in diabetic patients? Here are our option. It leads to hypercalcemia. It is expensive. It may cause allergic reactions. It can lead to a mutation in human genome. So before the discovery, or before before we invented the methods to synthesize insulin using E. coli through genetic engineering, back in the days, the cows and the pigs were slaughtered, and the insulin from these cows and pigs were used as a treatment for the diabetic patients. So we know that our body is highly sensitive for any antigen or any foreign particles that is coming from any source. So when it is coming from an alien animal for our body, so obviously it is a foreign animal or a foreign body. So when it is coming from that source, the proteins that are present in these animals are very sensitive for the human beings, and we start responding to it through allergic reactions. So basically, there was a lot of immunity based issues when it comes to the insulin extracted from these pigs and cows. So most of them was allergic reactions, and people, not everybody were compatible for this particular insulin. So that is why it was highly necessary to develop the insulin from our E. coli. Okay, so that is why our option C is correct for 181. So here we have 184 question. So we have to choose the correctly matched pairs and pairs and the correct option. So we have the first option, leptotin, chromosomes become invisible. Zygotin, pairing of homologous chromosomes take place. Pacatin, dissolution of synaptomial complex takes place. Diplotin, bivalent chromosomes appear as tetrads. Diakinesis, so terminalization of chiasmata takes place. So we have an option 1, 2 are correct, 1, 4 are correct, and 2, 5 are correct, 2 and 3 are correct. So this is actually an easy option, students. So basically, let's see. Leptotin. So we know that leptotin, zygotin, pacatin, diplotin, diakinesis are given in an order, especially for the prophase 1. Leptotin being the first stage in the meiosis 1, prophase 1, chromosomes become invisible. Chromosomes become visible when it comes to leptotin. Zygotin, pairing of homologous chromosomes takes place. So when it comes to zygotin, so this option, especially the pairing of homologous chromosomes takes place. Yes, that is also correct. So now what happened? It will lead to the tetrad formation, right? And the chiasmata. So now what happens in the next stage? Pacatin. Dissolution of synaptomial complex takes place. So pacatin is a stage where the important process called as recombination takes place, where the segments of the DNA are exchanged. So here is where the tetrad or the synaptomial complex is very prominent. So it is not dissolved. Okay. Next one. Bivalent chromosomes appear as tetrad. So here, in the diplotin stage, what happens? The tetrad actually separates and the homologous chromosomes become free. So, right? So this is where the dissolution happens. So that is why this option is also wrong. And coming to our diakinesis, thermalization of chiasmata takes place. So this is absolutely right. So the chiasmata terminates and the homologous chromosomes are completely free and they have finished their job. Okay. So that is why our 2 and 5 option are correct. So option C for 184 is correct. So we have the 192 questions. Select the correct options in the and you fill in the blanks. Okay. So when it comes to such a lengthy question, you go for one question after another question. So let's see what is the question here. So DASH is an enzyme that is crucial for immune system function, and its absence may cause to deletion of a gene. So whenever we talk about genetic engineering, we heard about something called as adenosine deaminase or ADA, right? Or ADA deficiency. So adenosine deaminase is a factor that is basically boosts the immune system in humans. So the deletion of this enzyme leads to a lot of immune compromise, right? So acute immunodeficiency it leads to, right? So that is why ADA. That is first blank is ADA should be the answer. Insulin consists of what? Dash and dash, and are linked together by dash. So when we see the insulin molecule consists of two chains. Actually it's three chains. That is A, B, and C chain. That is in proinsulin. But in matured insulin, we know that it is consisting of A chain, and it consists of B chain, and these two chains are held by disulfide bridges. Okay, disulfide bridges. Okay, so this is the correct option. Next one. Transgenic mice are being used to test the safety of what? So basically we use transgenic mice for the testing of safety of polio vaccine that the kids usually take every year, right? So we use the same transgenic mice, okay, as a organism of experiment to check if they're safe enough for our human beings. Okay, clinical trials. Next one. Dash involves silencing of a specific mRNA due to the complementary double-stranded RNA molecule, okay, that binds and prevents to the translation of the mRNA. Melodica and incognitia in tobacco plant. So we talked about this, right? So whenever we talk about melodica and incognitia, we know that we use RNA interference technology, that is RNAi technology. So basically, just inserting this gene into the host, they produce this, alternate forms of this RNA, which would complement with the RNA of the what you tell nematode. So basically neutralizing the nematode. So that is why this technology, which is RNA interference technology, it is also called as gene silencing technology, okay, is used in tobacco plant. So it is RNAi technology. So where do we have ADA, A chain, B chain, disulfide, polio vaccine, and RNA? Okay, anti-adenosine diamine correct. A chain, B chain, disulfide bridges, polio vaccine, RNAi. So our option A is correct. So we need not go for the rest of the options. Okay. Androgen binding protein, ABP, okay, secreted by Sertoli cells. What's the function? Inhibits release of FSH from the anterior pituitary, concentrates testosterone in seminiferous tubules, prevents development of Mullerian duct, okay, provides nourishment to developing spermatozoa. Androgen binding protein secreted by Sertoli cells. So basically, if you see the hormone regulation in when it comes to spermatogenesis, we come across this ABP, okay, ABP. So ABP, which is secreted by the Sertoli cells. So what they do is they actually go and bind to the interstitial cells, and they make them release testosterone. So it helps in testosterone concentration in the seminiferous tubules. Okay. So here we have the 200 question and the final question of the question paper. DNA contains nucleobases, sugar, and phosphate. Removal of which of these from the DNA sample, a sample will not significantly affect the length of the DNA. Okay. And we have the options, that is nucleobases, sugar, phosphate, none of the above. All right, students, let's see what's the right option. So here we have one of the, oh, sorry, I will just try to, here we have, yeah, I was right. So here we have our pentose sugar, right? So in pentose sugar, here we have the first carbon atom, fifth carbon atom, third carbon atom. Here in the first carbon atom, we have the nitrogenous base. So here in the fifth carbon atom, we have phosphate, and the third carbon atom as OH. So when it comes to the next adjacent nucleotide, here again, we know that the sugar group and the fifth phosphate and the third OH is participating in creating a bond, right? And here again, we have the OH, and again, here we have the nitrogen. So now let us imagine that we eliminate any of this. So let's go with the nitrogenous base. So whenever we eliminate the nitrogenous base, is the length of the DNA affected? Definitely not. Why? Because the bases of the nitrogen bases project towards inside, and they are responsible for creating the bonds between the adjacent, that is the other DNA strand is there, right? So they establish this hydrogen bond between them to hold these two DNA strands together. Apart from that, the backbone of the DNA is made up of sugar and the nitrogen base. The sugar and the phosphate group. So in sugar, if you basically take either phosphate group or sugar, it will definitely collapse the DNA's length, right? So for example, let us say that I eliminate, okay, the 5 prime phosphate and the 3 prime. So here is a OH, okay? So here is a phosphate. So I eliminate phosphate. What will happen? So there won't be a proper way how it can make a bond or establish a bond. So that is why sugar and phosphate is very, very important for the length of the DNA. Okay. That is why it gives a very uniform and stacked nature of the DNA. Whereas nucleobases will not affect because they project towards inside and they do not participate in elongation or in determining the length of the DNA. So nucleobases is the right option. So for 200 question, option A is correct. Okay, all right, students, here we come to the end of our discussion. So if you have any additional questions that you feel that you have the difficulty, please inform this to your subject teachers and they will reach to us and we'll come back with the solution. Thank you so much. I'll see you later. Bye, students.